Welcome to the fascinating world of enzymes. Shown here is a substrate trying to find its enzyme for hydrolytic dechlorination. Enzymes are responsible for important biological processes, so by optimizing the reactions they catalyze, we're able to increase reaction rates for generation of biofuels, synthesis of drugs, and degradation of environmental pollutants. Many of these biological processes are carried out by multiple enzymes comprising a metabolic or reaction pathway. As you can see, there can be an increase in toxicity to the cells if reaction intermediates are allowed to float around. These pathways can be optimized to avoid toxicity and increase efficiency by spatially controlling placement of the enzymes and arranging them in a supramolecular assembly. Co-localizing these enzymes reduces the amount of time needed for the substrate to find the next enzyme in the pathway, protects unstable intermediates from solvents, and prevents diffusion of toxic intermediates. But most enzymatic pathways, especially artificial ones, do not self-assemble by themselves. So how do we get them to do that? We came up with the idea to create these assemblies using melded binding domains. Shown here is a PDZ domain and its respective peptide. With many binding domains available, we can select a binding domain appropriate for the problem at hand. For example, we can use domains that only bind when a trigger, such as a kinase, is present, giving us control over the formation of the assembly. In fact, we can take this a step further by making covalently binding domains and enzymes through the utilization of unnatural amino acids. In our prototype of this approach, we used fluorescent proteins as a surrogate for enzymes of a metabolic pathway. Using computational software called Rosetta, we created models of our designed proteins with binding domains inserted. Shown here is a PDZ domain being inserted into the red fluorescent protein. On the other end of this protein, we can insert a peptide that makes this bind tightly to another kind of fluorescent protein, GFP. We can then take these computer models and bring them into life by expressing them inside of bacteria and isolating them in a tube. We can apply a similar approach to actual enzymes, bringing them together using inserted domains and peptides to generate large nanoscale assemblies. These are efficient assembly lines where chemical transformations like degradation of toxic herbicides can happen efficiently and with minimal toxicity, unlike with free enzymes. We hope that this technology will help biodegradation and biosynthesis of important chemicals.